good morning. Okay, as we know that today we'll be having a pre-conference virtual workshop entitled Data Management and Analytics with R. Of course, we need a technique, to process the data. Okay, so this is where your data analytic knowledge must be in.
There is sufficient evidence to conclude that the students who do well in theoretical biology tests also do well in practical biology tests.
The practical and theoretical tests, they are directly related. Yeah, there is sufficient evidence to conclude that these two variables, the marks of the theoretical and the practical, are having direct relationship at the 5% level of significance. Okay.
Good morning, ladies and gentlemen. Before we start our conference today, let us review a few technical details. First, to help us recognize you for all participants, please take a moment to rename yourself with participant _ your university name _ your name. For all paper presenters, please rename yourself with presenter _ your track _ your university name _ your name. For moderators, please rename yourself with moderator _ your university name _ your name. And for all committees, please rename yourself with committee, your university name _ your name. Second, to reduce noise and sound problems, please mute your audio, but don't forget to turn on your video and use the virtual background. Third, you may want to drop your questions regarding the topics via the Zoom chat box. And last but not least, we want to remind you to fill the registration form on the link intip.in/attendance@CDS 2021.
Assalamualaikum warahmatullahi wabarakatuh. We are delighted to welcome you to the 6th International Conference on Soft Computing in Data Science 2021. First of all, we would like to introduce ourselves. My name is Maura.
I'm Sekar.
From the Department of Statistics, Institut Teknologi 10 November, will be the master of ceremony for today.
This conference aims to provide a platform for researchers and practitioners to share their research work and to create international research collaborations on leading-edge methods, and also addressing challenges, problems, and issues in soft computing, especially in data science and analytics. SCDS 2021 have invited great international and local keynote speakers who are academia or practitioners to share their knowledge and experience in the area of soft computing in data science. The theme for this year's conference is Science in Analytics, Harnessing Data and Simplifying Solutions.
The conference is organized by Institute for Big Data Analytics and Artificial Intelligence, University Teknologi Mara, with the collaboration of Faculty of Computer and Mathematical Science, University Teknologi Mara, Department of Statistics, Faculty of Science and Data Analytics, Institut Teknologi 10 November, Surabaya, and Research Nexus, University Teknologi Mara.
In collaboration with the University of Tennessee Knoxville, Big Data Institute, University of Oxford, Universitas Gadjah Mada, Kyushu Institute of Technology, Charles Sturt University, Chulalongkorn University, Liverpool John Moores University, Data Analytic and Collaborative Computing Group, University of Macau, Durban University of Technology, and World Federation Soft Computing. Also supported by the Prime Minister's Department, Department of Statistics Malaysia, Malaysia Digital Economy Corporation, Quantetics, and Women in AI. It is an honor for us to introduce and welcome the keynote speakers of the day. First, Assistant Professor Dr. Ubydul Haque from University of North Health Science Center, USA. Second, Associate Professor Dr. Simon Fong from University of Macau, China. Third, Professor Dr. Richard Milham from Durban University of Technology, South Africa. And last but not least, Associate Professor Dr. Aidan Doherty from Oxford University, UK. We welcome all the presenters and participants from all over the world. It's a privilege to have you here with us on the second day of the 6th International Conference in Soft Computing in Data Science 2021.
Ladies and gentlemen, without further ado, we are now continuing to the first keynote speech of the day, which will be delivered by Assistant Professor Dr. Ubydul Haque from University of North Texas Health Science Center, USA. The keynote speech will be moderated by Dr. Achmad Choiruddin from Institute Teknologi 10 November. Please, Dr. Ubydul Haque and Dr. Achmad Choiruddin, the time is yours.
Okay, thank you very much. First of all, I would like to confirm that my voice is clear, Maura and Ska.
All clear.
Thank you very much. Okay, ladies and gentlemen, so our first keynote speaker for today is Assistant Professor Ubydul Haque from University of North Texas Health Science Center, USA. Hi, Doctor. Good evening.
Good evening.
Yeah, I know that in US it is now probably a little bit late, and it is a very honor to have you in this conference, and we appreciate your time to join us in this conference. So, ladies and gentlemen, Dr. Ubydul Haque conducted his PhD in public health from the Nagasaki University in Japan. Prior to that, he finished his Master of Science in Geoinformatics from the Royal Institute of Technology, and also a Bachelor of Science from Khulna University, Bangladesh. Before joining at the University of North Texas Health Science Center, he did a postdoctoral research at the Johns Hopkins University and also joining as an assistant professor at Baldwin Wallace University. Dr. Haque has been involved with global health and community-based research centered on epidemiology, biostatistics, and spatial modeling. Today, he is going to talk about spatio-temporal dynamics of dengue, chikungunya, and Zika viruses in Mexico, a potential co-circulation problem. Without further ado, please welcome Dr. Ubydul Haque.
Thank you. Thank you very much for inviting me. Today, I will talk about spatio-temporal dynamics of dengue, chikungunya, and Zika virus in Mexico, that a potential co-circulation problem. As you know, those people who are living, especially in Southeast Asia, you are very familiar with the arbovirus. So dengue is a major arbovirus and a big health problem in Mexico. The three most important viruses transmitted by Aedes aegypti mosquito, including chikungunya, dengue, and Zika virus. These Aedes-borne viruses are considered human amplified urban arboviruses because humans play the major primary reservoir and facilitate this spread of this infectious disease. The mosquito thrives in urban areas where human disease burden is highest, although chikungunya and Zika are concerning. The exact global burden of disease is unknown. The incidence of dengue has increased dramatically worldwide in recent decades, and over the last 2 decades, the number of dengue cases increased over 8-fold, and nearly 105 million dengue virus infections have been reported globally each year. The Latin America alone, reported nearly 16% of global dengue burden. And this is the map of spatial distribution of arboviruses in Mexico that reported from 2012 to 2019. And this is the aggregated data from dengue, chikungunya, and Zika, and then we plotted this data at the municipality level. This is the aggregated data over the last 8 years, and we categorized with 5 categories, very low to very high. And you can see the red hot spots, especially that is very high. That is parts of the Mexico is reported the highest endemicity. But we want to know exactly why, what is the reason, what is the risk factors behind that. That is started from because we all know that a special arbovirus that is multifactorial, that is not a single reason. That's why, for example, climate may have some role, socioeconomics may have some role. So we are going to investigate. So at the very beginning of the study, we try to explore the pattern exactly. So in a more better way, you can see exactly how the special clusters are. This we use Satiscan software to detect these special clusters here. So we detected 19 geographic clusters where dengue, chikungunya, and Zika actually have reported higher cases compared with the other neighboring municipalities. As I said, that this actually reported at the municipality level. So we aggregated and analyzed that at the municipality level. So next, please. 21 statistically significant clusters were observed in Mexico. We analyzed for all clusters and non-clusters, as socioeconomic features and climate features may have different levels of impact for all clusters and non-clusters. We use 3 approaches: Pearson correlation coefficient, randomized dependence coefficient, and Shapley additive explanations to compute the weights of socioeconomic features and climate features that actually that is associated with every geographic cluster. That means the 21 clusters that we detected here. Here, the majority vote means if 2 of them indicate one has more weight than the other. That means it has multiple socioeconomic risk factors that we combine together, and as well as it has the climatic factors like mean temperature, mean maximum and minimum temperature, rainfall, humidity. So if more than 2 of them indicate that one has more weight than the other, then we compute the average value and those 2 values as the final weight. So if 3 of them are in the same direction, all indicate one is more important than the other, then we take the average, means we take average value for these 3 approaches. Here it indicates the majority vote and average results for chikungunya, dengue, and Zika prevalence based on different clusters. There are 3 spatio-temporal clusters for chikungunya and 12 spatio-temporal clusters for the dengue prevalence, and climate features had more impact than socioeconomic features on model outputs in clusters 1, 3, 4, 5, 6, 7, and 12. There are 6 spatio-temporal clusters of Zika prevalence, and climate features had more impact in cluster number 1, 2, and 3, whereas socioeconomic features had more impact than climate features on model output in cluster number 4, 5, and 6. Can you please go to the next? So we use the Pearson correlation coefficient, randomized dependence coefficient, and Shapley additive explanations to analyze the influence of socio-demographic and climatic factors on this prevalence. So Shapley additive explanations is a method to explain individual predictions. The goal of Shapley is to explain the prediction of an insane by computing the contribution of each feature to the prediction. And Shapley values tell us how to fairly distribute the prediction among the features. A player can be individual feature, or a player can be also a group of feature values. The idea behind Shapley feature importance is simple. Features with large absolute Shapley values are important, and randomized dependence coefficient is a measure of nonlinear dependence between 2 variables. So here you can see the weights of socioeconomic features and climate features by using Shapley, RDC, and Pearson approaches. And you can see exactly that where climate have more contributions, or where socioeconomic have more contributions for all these 12 clusters, especially for the dengue prevalence. Can you please go to the next? This table shows the weights of socioeconomic features and climate features by using Sharp, RDC, and Pearson approaches, especially for the Zika prevalence. Here you can exactly see that socioeconomic features and climate features, they have more impact in all 6 geographic clusters for the Zika infection. Please go to the next. No, the previous one, please. Okay, and this Yeah, right. The weights of socioeconomic features and climate features by using Sharp, Ardis, and Pearson approaches are presented here. For the chikungunya infection, they had also 3 clusters. And these clusters, you can see exactly that what is the contribution of each feature, either socioeconomic parameter, either socioeconomic or climate for this for the geographic clusters and the association with them. Next, please. Here I presented 2 figures. One is the figure A is the average impact on model output magnitude for chikungunya all clusters, and the second one is that the figure B is showing average impact on model output on magnitude for chikungunya for non-clusters. As you can see in the previous map that I showed you, 3 maps presented with yellow figure that showed that there is 3 clusters for the chikungunya infection, and you can see exactly that. So that means I cannot exactly see that there are 2,511 municipalities in Mexico. So these 3 clusters, they probably presented by with 100 municipalities. So that means that the rest 2,400 municipalities, those are considered as non-clustered, but those with that reported the higher number of cases compared with the other municipalities, and those are statistically significant. Those are considered as geographic clusters. So you can see exactly that. So here we also try to understand what is the contribution of each parameter that I think you are trying. You can see that literacy rate, population without health services, houses with floor of darts. So all these, many of them are socioeconomic parameters. And from mean maximum temperature, minimum temperature to minimum rainfall millimeter per day, those are the climatic parameters, and we also the altitude, because we know that actually temperature varied up to with altitude. So you can see from this that we set a threshold for the infected prevalence. If the number of infected prevalence of a specific instance exceed the threshold, we defined it as a class 0, which represented the infected class. Otherwise, we defined it as class 1, which represents the normal class. For all 3 arbovirus diseases, the influence of socioeconomic attributes was larger than the influence of climate attributes for some clusters. And for example, we found that socioeconomic attributes had a higher impact than climate attributes. And the weighted socioeconomic attributes for Shapley value here is 0.61, and the weighted climate attributes for Shapley value here is 0.39. Here we set a threshold for the infected prevalence for a specific instance. It contains information, location, infected prevalence, etc. Go to the next, please. Here we are presenting average impact on model output of magnitude for all dengue virus for all clusters, all 12 geographic clusters, and average impact on model output for magnitude for dengue virus for non-clusters. And for all 3 arbovirus diseases, and then for all 3 dengue virus, the influence of socioeconomic attributes was also larger than the influence of climate attributes for some geographic clusters there. And we found here that the attributes of altitude and minimum rainfall had a marginal influence on the model output, whereas the average rainfall volume and the maximum rainfall volume are more important than the minimum rainfall volume concerning the model output for the rainfall volume. Thank you. Next one, please. And these 2 figures are presenting Zika, where that means the average impact of model output for the Zika for all clusters, and the average impact on model output for Zika for non-geographic clusters. The weighted socioeconomic attributes for Shapley value here is 0.61, and the weighted climate attributes Sharp value is 0.39 here. Here we also found that attributes of altitude and minimum rainfall volume had a marginal influence on the model output. The average rainfall volume and the maximum rainfall volume are more important than the minimum rainfall volume concerning the model output for the rainfall volume. In the spread of Zika infections and at the same time the geographic clusters of Zika infections in Mexico. Next one, please. This is the classification performance of different machine learning algorithms on dengue, Zika, and Chikungunya virus predictions for all geographic clusters. So here all three infections are together. So the figure is a little bit crowded, but I hope you can see that. Here I'm trying to display the performance of different machine learning classification algorithms across all geographic clusters after taking the average. The F1 scores of XGBoost for Chikungunya, Dengue, and Zika prevalence are larger than other baseline approaches, which means XGBoost has better performance than other approaches. For instance, the accuracy of XGBoost under dengue prevalence is 0.87, which is higher than the corresponding weighted accuracy of 0.78 and precision 0.85. This may happen due to class imbalance issue, and more specifically concerning normal prevalence, which is called normal class, and we had low prevalence there. Next. This table is representing the standard error of classification performance of different machine learning algorithms on dengue, Zika, and Chikungunya virus prediction for all geographic clusters. Here you can see the standard error of classification performance on Chikungunya, dengue, and Zika prevalence. And for example, the standard error of XGBoost under dengue prevalence for accuracy, weighted accuracy, precision, recall, and F1 score were 0.7, 0.8, 0.13, 0.07, and 0.09 respectively, which are lower than the most of the standard errors of the other baseline approaches. Next one, please. This table shows the performance of different machine learning algorithms across all non-geographic clusters. Earlier, I presented geographic clusters, but these are non-geographic clusters. After taking the average, the results show that XGBoost performed the best in terms of precision measures for Chikungunya, dengue, and Zika prevalence. And the values of accuracy are larger than weighted accuracy and precision values. Next one, please. This table indicates the standard error of classification performance of different machine learning algorithms on Chikungunya, dengue, and Zika case predictions for non-clusters, and the standard error of all clusters and non-cluster predictive results in accuracy, weighted accuracy, precision, recall, and F1 measure. XGBoost performed the best for Chikungunya, dengue, and Zika prevalence, and the standard error of XGBoost is more stable than other baseline approaches. The next one, please. The cross validation can overcome the overfitting problem. We split data into 10 non-overlapping subsets. Each time we use one subset as a testing set and use the rest data as a training set. We repeat the process 10 times by taking a different training set and test set. We take the average result as the final result, and the model was evaluated based on classification accuracy, weighted accuracy, precision, recall, and F1 score. Based on the results presented on the previous tables, the accuracy of different approaches is higher than the corresponding weighted accuracy, precision, recall, and F1 score. The next one, please. There are some limitations in this study. The value of accuracy were higher than the weighted accuracy and precision values, possibly due to the imbalance scenarios. And compared to the normal cases, we had fewer cases of infection in some geographic clusters. And in many municipalities, they did not have any cases. So the accuracy only considered the correct predicted cases and not which class the case came from. Therefore, if the data set is imbalanced, we need to add more evaluation metrics to evaluate the model performance. In our results, we used weighted accuracy, precision, recall, and F1 score to evaluate our models. Here we used aggregated data at the municipality level. That means this is not the individual data, or this is not based on the household. So this is the combined data that we aggregated together from the all cases for each municipality. This study also used only laboratory confirmed cases and potentially lost many positive cases. The risk factors for arbovirus transmission are also determined based on passive surveillance system. In this study, we also used municipality level data and adjusted it for population density. And although the municipality level data has been widely used, it could be possible that some of the observed patterns are confounded by potential hidden factors in our data. as many factors, for example, rural areas that we did not adjust yet, but we're trying to bring more additional variables to rerun the analysis again that may also influence the distribution of Aedes aegypti, and identifying and addressing these hidden factors could be of great interest for any future studies. The next one, please. Yes, the distribution of parvovirus infections is often driven by local spatio-temporal patterns influenced by fine scale, socioeconomic, environmental, biological, and demographic factors. And the current analysis at the municipality scale is too crude to capture many of these drivers of transmission heterogeneity. This implies a clear need for the development of a more integrated individual and household level with fine time scale data to understand the implications of these household patterns for targeted disease surveillance and vector control activities. However, we actually analyzed nearly 250,000 positive cases of dengue, Zika, and Chikungunya cases in Mexico over the last 8 years. That means that's a very longer time series data, and the sample size is very big. And for any future study should be used to build predictive models to anticipate the time and location of parvovirus outbreaks and determine the risk factors, incorporating microclimate data, landscape ecology, urban environment into disease transmission models. That has the potential to predict and ecologically more interpretable metrics of mosquito-borne disease transmission risks in urban landscape especially. And further study of disease clusters concurrent with entomological data on Aedes distribution and human contact would be also beneficial. And a better understanding of the drivers of arbovirus transmission that consider local dynamics should contribute to the design of a more effective vector control program, disease prevention, and promote public health in Mexico and other endemic countries. The next, please. Thank you.
Okay, thank you very much, Dr. Haque, for sharing your research. We have plenty of time for discussion, so I would like to invite all the participants who has any questions, comments, or remark. You can use the feature that we have here in the Zoom by using the raising hand feature, or you can also type your questions in chat column. Actually, I have one question, Dr. Haque, about this. So you say that actually before doing the classification, you try to use kind of the exploring the pattern and you find kind of some clusters on that. When you try to make the cluster, do you also consider like the spatial dependence between locations on that?
We have 2,511 municipalities. So we created centroid in GIS, Geographic Information System. And based on the centroid, we linked exactly the total population for each municipality and total number of cases per year in each municipality. So from there, we run the StatScan. That means we adjusted population denominator when we try to identify the spatio-temporal clusters. Is that
okay.
Your question?
Yes, it is. Yes, exactly. Okay, thank you. I would like to invite other participants, or if you have any other questions or comments or remarks, please.
Morning.
Oh, hi,
Morning, Dr. Ubydul. Hi, Dr. Ahmad. Can I ask Dr. Ubydul some questions?
Yes.
Hi, Dr. Ubydul. Good to see you.
Good to see you too.
Okay. You did the clustering, right? I have a few questions. What method do you use to do the clustering?
Sorry, please.
What method do you use to do the clustering?
I use StatScan first to detect the geographic clusters.
Okay.
There is a software that is called StatScan that can run the spatio-temporal clusters based on the location and space. So if you just give them the latitude and longitude of each cases, for example, we can use that based on the household location, based on the individual's location, or based on the municipality. Since we use the aggregated data, that means we compile the data for whole municipality, and we had 2,511 municipalities here in the Mexico, and we have the geographic polygon. So polygon boundary for each municipality. So we created the centroid of each municipality first. And then when we run the spatio-temporal clusters, we showed the software of this 2,511 locations of centers of each municipality, and we gave them the monthly number of aggregated cases for all 3 different infections separately, and that software, and then we also gave them the total number of populations. So it adjusted population, it adjusted time, and it adjusted space. And it identified 12 geographic clusters for dengue, 6 for chikungunya, 6 for Zika, and 3 for chikungunya. 3 for chikungunya. And those are statistically significant, highly significant, with 99% confidence interval.
So what do you mean in your data when you have clusters and non-clusters?
Clusters means those municipalities that reported the significantly higher number of cases, even after adjusting the population compared with this neighboring municipality.
So when you did the bar chart, right? You did for all when you did the bar chart, you have all clusters and non-clusters. I'm not very clear with that results.
Okay, so it only detected 12 clusters for dengue virus. For that means all 3 infections out of 2,511 municipalities. I think 1,500 municipalities that came within clusters, and rest of the municipalities that did not report, that did not come within clusters. And I did not show the map, but if you see them, my previous publications, it shows that many of the municipalities, for example, 1,400 municipalities, they never reported Aedes mosquitoes, because some of the municipalities in Mexico, they are in very high altitude, so mosquito cannot survive there. So many of the locations are desert, so they don't have water. There are no human settlements. So those are the municipalities that did not report any infections.
Okay, Dr. Ahmad, can you show the bar chart, please? You talk about social and weather data. How can you conclude? What are the social indicators that affect these diseases? Dr. Ahmad, can you show the slide on bar chart?
Yes, the committee will help me as well.
Thank
for this
you.
One. The bar chart, Mas Tantan.
Yes, because he showed the social indicator and the weather indicators together.
Yeah.
So I would like to know what are the social indicators that affect these viruses?
Okay, here you can see that literacy rate, that had some indicator. It is also aggregated at the municipality level, that how much, for example, 20% people don't have education in Mexico, in that particular municipality or some municipality. And population without health services, that means they don't have proper hospital. Or for example, they have the limited number of hospital. That means 20% people have access to hospital in that particular municipality. And houses with floor of dirt, that means most of the people are living under poverty in those particular municipalities. Others are houses without toilet. That means, for example, 10% people don't have the running toilet in those municipalities. And others are houses without water pipelines. That means that is a certain portion of that population in that particular municipality. They don't have sewage, they don't have electricity, and these are the socioeconomic parameters that came with significant predictor for those particular municipality. And other climatic parameters are mean maximum temperature, mean minimum temperature, mean rainfall per day, and maximum rainfall per day, and minimum rainfall per day. But that has very low impact, and altitude doesn't have much impact.
So your class 1 is municipality that has chikungunya.
The first one is for chikungunya, right?
And then 0 is without.
No.
The area has no.
Here, this is the threshold we defined. For example, if the number of infected prevalence of a specific instance exceed that threshold, we defined it as class 0. That means if it is more than 0, then actually we defined right. If it is less than 0.2, If it is less than 0.2 for here, no. Class 0 means which presented the infected class, and others were defined at class 1, which represent the normal class. That means it doesn't have much impact. It has more impact. One has more impact than the others. For example, here I will say that population without health services has much impact on the chikungunya clusters compared with the non-clusters.
Because if you see this chart for literacy, is higher for class 1. So I'm trying to interpret what does it mean.
Class 1 means it is the positive clusters, and class 0 means the negative clusters.
All right. And you have standardized all these variables to be the same range? Because I'm sure weather and social indicators are different values, right?
Right.
So you have standardized them, right?
Right.
Okay. Thank you so much.
You're welcome.
Thank you very much, Prof. Yep. So we still have probably one or more, two more questions or remarks. If anybody from the room have any questions, remark, or comment, please. I would like to see in the chat column if you have any.
Only for one question, please.
Please.
May I ask
Sure.
a couple of questions, Professor Haque? Yes. All right. Zika virus, I think I live in Singapore before, and this outbreak usually happens often in the same places, at the same places, so-called hotspots, almost every summer, especially after the rain. So my question is, how would you take advantage of knowing those hotspots in your analytics and your result? And my second question is, you have done great job in analyzing the data. How would you connect the result of your analysis to the maybe a medical authority on maybe preventing, or subsiding that Zika virus?
Thank you. I will appreciate if you. I understand your second question, but if you please repeat your first question again, that will help me probably to try to answer as much as I can.
All right, I'm sorry. The first question is, Zika virus or Zika's disease usually happen at certain hotspots. For example, in Singapore, we know that every summer there will be about at least 3 or 4 hotspots in outbreaks that always have disease outbreak. So the question is, how this knowledge that where are the hotspots that usually happen will contribute to your data analysis? So that's my first questions.
Thank you. Okay. So we had data from 2012 to 2019. So to answer your question, I need to go back and check exactly that yearly hotspots. So I agree with you, but I don't have much idea about the Zika infections. That means I did not analyze in that way so that I can directly answer your questions. But yeah, that is true that it might be the geographic, that means that it can be a persistent hotspot, or you can say that it is a stable hotspot. So if that is true in that case, but then the data I did not explore in that way. So we thoroughly analyze the data, but if that is the way, that means that in terms of this analysis, our goal is to identify the either climate or socioeconomic. If socioeconomic, which parameters, and if climate, which parameter that contributed more. So you know that for the climate impact, the parts of the world that are going to pay more price and that going to be more prone of the global warming associated climate change. So part of the Mexico have diversified climates. Some are have a huge rainfall, some are arid, some are deserts, some are mild weather. So considering that you are true that the eastern part of Mexico that have the high rainfall that came with persistent hotspot. Another second hotspot if you see in the western part of Mexico that is called Colima, where actually we're trying to set up a planning for a longitudinal study. So there is a stable hotspot there. That is true. I agree with you. And at the same time, we try to identify what is the role of the socioeconomic and what is the role of the climatic parameters. So you can see it is almost 50-50 in some clusters, somewhat 65-34. That's why we are planning for an in-depth longitudinal study to understand at individual and household level. So how actually it is going to impact both socioeconomic, individual, or the health from the health seeking behavior perspective, or from the genetics perspective. That will probably give more in-depth analysis, in-depth and a lot of answers that are as well as the stereotype. And your second, right? I understand your second point of question that how actually we can put it for the control perspective. I'm not ready to say how actually for the doctors or from the what I call from the others part, but I can tell you that these maps and the result we produced at the municipality level, we are ready to share with the Mexican government and Mexican Ministry of Health so that they understand that which municipality they have the high infections, which are hotspot, where actually the special predictor like socioeconomic, different socioeconomic parameters, different climatic parameters, so that what they can understand what they need to go for enhance the surveillance system, take more control efforts, or actually they need to put more medical efforts. So these are the informations, these are the decisions the Mexican Ministry of Health can take from this study. And this is also a very large data set that I did not see any other study that published nearly 250,000 positive cases. Those are lab confirmed cases together. I know Malaysia have data, but Malaysia did not publish that data. So this is the information that the Mexican Ministry of Health, they can use it for the control perspective.
Thank you very much, Professor Haque. We learned a lot. I don't know. I think that maybe by knowing the hotspots, hopefully the predictions will be more accurate, and the medical personnel would be maybe better prepared when they have the insight from your analysis, or maybe even forecasts.
Thank you.
Thank you. Thank you very much.
Okay, thank you very much. If there are any questions or short remarks from the participants? Okay, so if not, please let's thank Dr. Haque and all the participants for a very fruitful discussions. We hope that all the participants can benefit from this talk, and we greatly appreciate Dr. Haque for your time and your effort to join our conference here, and we hope to see you again soon.
Thank you.
Okay, so probably I will get this back to the MC.
Thank you to Dr. Ubydul Haque and Dr. Achmad Choiruddin. As a token of gratitude, we would like to invite Dr. Ubydul Haque to receive a certificate of appreciations. We greatly appreciate your time you spent with us today, and everyone, please give us your thumbs up, your reactions through the Zoom. Thank you. Professor Ubydul Haque. Yeah. Thank you, Professor. And now, ladies and gentlemen, let us welcome the next keynote speaker, Associate Professor Dr. Simon Fong from University of Macau, China. The keynote speech will be moderated by Dr.rer.pol Dedy from Institute Teknologi 10 November. And ladies and gentlemen, please give a warm welcome to Dr. Simon Fong and Dr.rer.pol Dedy.
Hello. Hello, Prof.
Yes, I'm here.
Simon Fong. Okay.
Could you see the PowerPoint?
Yes, we can see your PowerPoint clearly.
there would
Okay.
be some maybe mini videos along the way. So I
Okay.
hope the
Okay.
bandwidth is good.
Okay. Okay.
All right.
I'm the moderator. Before you start your presentation, I will give some introduction for you, and yeah, we can start the presentation afterward. Okay. Thank you for the MC Sekar and Maura. Good morning, everyone. Respectable the conference chair, Dr. Jerry Purnomo, Professor Yapdiwah, Honorable all speaker, committee and participants, Associate Professor Dr. Simon Fong, graduated from La Trobe University, Australia, with a first-class honor, Bachelor of Engineering in Computer System degree, and also a PhD, a Computer Science degree in 1993 and 1998, respectively. Now, Professor Simon is working as an associate professor at the Computer and Information Science Department of, Computer and Information Science Department of the University of Macau. He is also one of the founding members of the Data Analytic and Collaborative Computing Research Group in the Faculty of Science and Technology. Before he joined the University of Macau, he worked as an assistant professor in the School of Computer Engineering, Nanyang Technological University, Singapore. And prior to his academic career, Prof. Simon took up various managerial and technical posts, such as system engineer, IT consultant, and e-commerce director in Melbourne, Hong Kong, and Singapore. Some companies that he worked before include the Hong Kong Telecom, Singapore Network Services, IAS Pro Data, and United Overseas Bank, Singapore. Associate Professor Fong has a career as IT practitioner and professorship over 14 years. He has published over 288 international conference and journal papers, mostly in the area of data mining and optimization. Okay, without any further ado, now allow me to welcome the keynote speaker, Associate Professor Dr. Simon Fong, to deliver the presentation. We have 45 minutes, including question and answer. The time is yours, please.
Thank you very much, Professor Dedy. Thank you, the Committee of SCDS 2021. Thank you very much, University of Technology Maura. Very honored that I could be here again to share some work with everybody. Now, the topic that I bring up today may be slightly different from the brochure, because I think this is a popular topic that actually many research teams or researchers want to overcome. That is early cancer detection. All right. I am basically now not in Macau. I am in Tsinghua University in Beijing, having my research leave. So whatever I'm showing is the latest. And I am bringing up basically a sort of open questions. That is how best we can do in early cancer detection, which is never an easy thing. Now, I just show some pictures. This is my PhD students and me, and we are with a electronic microscope that has one of the highest resolutions in the world, into the nanometer scales. Usually it's microns if we're talking about MRI, CT scans. We have the hardware that can look into nano. So the next question is, are we able to see at the level of genes and DNA? If we could, how this, genes or identification of genes would relate to a kind of cancer, specific cancer for specific patients. I think you may have heard that we could have test the DNA in order to know in advance the possibility of whether somebody would have a cancer or not, the livelihood. So we are moving along this way. What I am responsible doing, let me just show you a bit of background. I am on I won't say the most deadliest, but it's the most dangerous cancer disease. Why? It is not ranked as number one, like heart disease or lung cancers, which are um, the most deadly ones, because that it is extremely difficult to detect it at the early stage. Most people may have mistaken as gastritis, that means inflammations or maybe ulcer, maybe irregular diet and so on. And as you can see in this chart here, the survival rate actually dropped below half once we miss out stage 1. Okay, now before we move on into the DNA thing, we have another team doing the DNA, the mapping the genome data to the cancer data. But my team, basically me and my PhD student, is very focused on to pushing the highest possible accuracy. Even 99% is not good enough, because we have 1% chance that we may miss out somebody, maybe a false negative, for example, this person is actually in stage 2, but we are telling him or her that, it was okay. And then the chance of survival actually drops. And it is actually quite related to us, because as you can see that this kind of cancer, when compared to the bottom, even in Africa or Europe or United States, right, more likely it will happen in Asia somehow. I don't know why. We're still trying to figure out why. Now, just a little bit of background. I believe that many of us are doing deep learnings on visual images. So usually to detect whether there's cancer or no cancer, you can have a scan of the organ. But in that case here, we are taking a small samples anyway, from the stomach wall, and put it under the microscope. So traditionally, we have the data mining that is put into these few main categories. That is pre-processing, feature extractions, segmentations, and classifications, and so forth. Now, the question is, if at a very early stage, it doesn't make sense to take a sample from someone. You know what I mean? Like at a very, like for example, I have the pain here and it just persists for a few days. We may not, go into the extent of, having a microsurgery, take a sample, this is considered as intrusive. So we are looking at something non-intrusive, which is the endoscope, which is a little fiber optics with a little camera attached that go all the way down to us. So we're dealing with visuals like in this picture here. Right? So that is actually the challenge, because traditionally, if we do a classification on a sample like this, that would be simple. Even bionic eyes, you can see that the little tiny dots there, they are the tumor cells. All right, and it's a matter of estimating how many tumor cells are there, how big they are. But we are not doing something like this. We are dealing with images. I'm sorry, this is in Chinese, because in 2018, about 3 years ago, one of the research teams in China has published this in a Chinese journal anyway, using 19 years to try to predict or classify whether it's cancer or no cancer. So put it in English anyway, we're comparing the AI, which can achieve about 88% specificity. Accuracy is 89.4%. Compared to human naked eyes, because through the little camera, you can see the images, not high resolution anyway, it's pretty low resolutions. So compared to humans, the first two are the junior doctors, and the last two are the senior doctors. However, the accuracy is not high, just about 70%, 60%, but by using AI, there's 89, which is not bad, it's promising. So we have hope. And at the same time, same year, 2018, the Japanese team, I think it's from Tokyo hospitals, they are also using convolutional neural network and trying to do the same thing. That is based on the images being captured as a video, and in real time, try to figure out what it is. But they were a bit lucky, because they use Haque, which is a very good I would say is more mature. Like GitHub, for example, there's a lot of software tools, are available on the platform. So Cafe at that year was one of the pioneers. So the result was quite good. In that Japanese team, still point out that there's still something not very perfect. Let me just quickly show you why with this example here. Now, those with green boxes are the ground truth. They are marked by the doctor. By just naked eyes, it's very hard to tell, except maybe this one here is quite obvious, there's some red mark. But the one in the center is almost not visible, all right? But when look closely, the doctor will be able to tell that, yeah, there is actually a sign that, this person is having an early cancer. And the photo below, as you can see here, the yellow one is actually a false positive. They're the reflection of the light, but it's taken as something unusual, in the pictures. On the other hand, this is also the result from the Japanese team in 2008.
Họ lành hóa cái khuy cái chia sạch cái chả mà chỉ cái máy quá là ngõ mà hay mày mày.
Okay, thank you. In this picture here, you can see there's a lot of false positive due to, I don't know, many reasons. What makes it so difficult is the whole thing is dynamic, because when you're, trying to get the best visual, you're moving the camera and the stomach chamber also moving. So the view is moving and the object, they are both moving anyway, and there's a lot of reflections. And whatever done there, it's hardly to get a very good still picture compared to maybe X-ray or MRI. So that is a very challenging work. And this is the kind of, stress that I'm facing nowadays, every day. All right, 2021, which is just this year, in Lancet, incredible. Okay, I mean, the journal is incredible, but the work itself, it was not absolutely difficult if you look at it carefully. This is in real time anyway, and they are trying to detect what is normal or abnormal in real time, which you can actually pull the video clip into frames and, frame by frame. So it has to be fast, it has to be good and accurate. I think these are the requirements. So the technology is not extremely difficult. Let me just move on a bit. Also, 2019, the Japanese team also were doing something similar in real time. So as you can see that all they need is just a normal PC with good GPU, capturing device, which may be a hardware interface, a driver, and the AI actually is sitting on the PC to real time diagnoses what is happening. And if you look at the hardware, it's pretty old actually. It's GTX 1080. All right, they need 2 cards anyway for training and testing the model. So I just want to say that it is affordable and is feasible for many hospitals and many clinics. So how we could improve the algorithms perspective, how we can improve the AI, until maximum. This is some screen capture once again from the Japanese team. As you can see that not only they detect yes or no, but they also will try to identify the type of cancer, because actually gastric cancer is different from the rest. There are 4 main different categories, and there are subcategories, subcategory, and so on. So overall, there are more than 20 so-called class labels if you're talking about classification. Now, this is what we did as something very preliminary. This is a small video anyway. I believe that is not difficult. It's just using OpenCV, using ULO, and we just train a bunch of pictures with the answers, with these problem images, and we also trained the deep learning models with those normal. So we have the data for abnormal and normal, and we just let it run for 12 hours using Google Colab. I think many of you may have done it before, right? Between cat and dog, or apple and banana. So same logic here, but what you can see here is, you can see the accuracy is not high. That is one thing. And the second thing is, it could never be precise. Like in this picture here, instead of telling me that there are blood, there are some polyps. Polyps means there's a lump. And also the white part, which is the ulcer. There are actually 3 different ones, 3 in 1. But you hardly can zoom in and to accurately identify it. we're still trying. So this is the kind of problem that we are facing right now. And just to give you a kind of feeling. The three main challenges that we have seen from the literature review is we are not dealing with histological data. Histological means it's a still picture of the tissue, all right, maybe a lung or maybe a brain, whatever organs. But we are dealing with endoscopic that is dynamic, and you just seen it is keep moving, and the light reflection, the
Professor Fong, maybe to help the streaming get better, you can turn off your camera. Yeah. Okay, please. We cannot hear your voice. Yeah. Yeah, we still cannot hear your voice.
all right. We got
Okay.
four main type of problem: polyps, which is a big lump, and we have this rough mucosa, which is a surface, and also we have lesions. Lesion is a bit tricky. That is something not
Sorry, Prof. Fong, we cannot hear your voice. Yeah. Can you hear my voice? I'm sorry, I cannot hear your voice.
Recording stopped. Recording in progress.
I'm too far away. Can you hear me?
Yes, we can hear you now. Thank you.
All right. Yes, it sort of comes on and off anyway. But thanks for telling me. How the image was turned into into mask. The black and white are the masks anyway. In this way here. So we use image processing, and next I will show that we will use the clustering as well. Data either is 0 or 255, but in this case it's red. We just sort of put it. So somehow by looking at these so-called river patterns, by applying the red mask, actually we know that it's a red color. So this is some of the this is how it will be converted into binary information, which can be the ingredient for training a neural network, right? Let me see what else. This is another example anyway. I'm sorry for this plot thing. I know it's a bit gross. Hope you don't mind before lunch. Okay, maybe those under 18s close the eye for a while. But I mean, scientifically, this is how we do it. We apply the filter, and then we convert it into just 2 color, all right, to represent the blood. And you can see the corresponding pattern. This is how we train the neural network instead of using the full images. What else? I think I'm almost done here. That actually shows another way of applying the mask. It's called dilate and erupt. I think it's quite famous in image processing. What you see is actually a manual work, but we wrote a script so that it will be automated. And our objective is to mark the boundary so that it will look as closely resemble to the real thing as possible, which again is not a very easy thing. All right, so you can see that we are fine-tuning that anyway. If it's too small, it may miss out the information, but if it's too big, for example, it may lead to some false positive or false negative. So this is quite a challenging thing. Anyway, now the other thing is we zoom out, all right, because we look at those Japanese team, quite a few cases of false negative missing out. They are due to the resolution, all right. So those zoom out pictures would also need to be take care. So I think I have a video for that. I think this shows about it's about how the bright spot could be removed by using the algorithms. But of course, it's manual. We have to write a script to automate this process. And anyway, so this is the way. I mean, just move on a bit quicker. Now, in this example here, we are using grids to locate where it is. So in terms of training the deep learning models, they are actually two steps. First is Euro. We identify somewhere suspicious, and then we use the grid, we zoom in, we will see this by going through this processing filter. Last one, which is the red spot detection. So in the red spot detections, which is quite easy anyway, because the blood is a very distinctive color. All right, so I just so that it could be put into a filter. I guess this should be the last one here. All right, so we mark the boundary as well, and we use these informations for training. This shows what it is. Okay, let me just move on into the last part here. Now we tried the other information as well. The one on the most left is the height. All right, because when you look at the thing, it's actually 3D. The object is 3D. It's not just a picture. So we have the spatial informations, and also we have the height information as well, for locating somewhere that is suspicious. So the features that we use, we sort of fuse them. This is the most basic way anyway, from the two ingredients. One is from the images, but instead of using the full images, we use the image mask informations. And also we look at the texture, how smooth it is or how rough it is. And there are two algorithms which are very classical. One is called grade level run length matrix, GLRLM. The other is GLCM, the grade level co-occurrence matrices. So we use these informations together with the images and to try to train the deep learning models in such a way. This actually shows about how the GLCM were obtained or extract from the pictures. We wrote a MATLAB script for extracting these. So for GLCM, which are the textures, we have 22 features. These are standard and has been published. Surprisingly, it was published in 1973. It's a very classical algorithms, and I think so in 1999 use it again for extracting the roughness or the smoothness texture information. All right. So we did some experiment. The good news is, if you look at this histogram, the values are distinctive. You get different values anyway, for different histogram features. So in other words, we are testing how useful the features are. Now, if you look at each individually GLCM, you can see that the difference between red and blue, especially the first one, F4, all right, you can see that they are quite distinctive. When they are the same, it's not good. When they're the same or very similar, it's very hard to tell them apart. But the good news is not bad in terms of medians and the interquartile range, they are quite different. So it just shows that not every feature is good. Like this one is good, F5, very distinctive. These are good as well. I should F10 maybe not very good, or F11, they are about the same. The blue and the red is difficult anyway. Not possible, but it's difficult as a feature. So we test it with all the 22 features in such a way. And after that, we use the other classical algorithm called GLRM. These are another classical algorithms that will convert the image into features. In this case, we have 11 features. SRE stands for short run emphasis, long run emphasis, GLN, and so on. the advantage, as you can see, that the more feature extraction result that we use, if they are relevant, I hope that they would give a good model. Otherwise, it will become overfitting. We didn't run any feature selection so far, but we just try to experiment about what is the best way of doing the deep learning. And once again, as you can see, these two butterfly plots, they are quite distinctive between the, especially RP here. LGRE, not very, they are about the same. But, some of these are very good. So as a conclusion, not yet, I mean, as a summary, this is what we propose to do. We use this ingredient, this information from the mask, from the segmentations. Watershed is a very famous classical algorithms anyway to mark the boundary so that we know what it is. So we take this boundary segmentations informations and also the texture information as number 3 and number 4. And we try on something so-called the latest. What latest now is DeepLab version 3. I think it just came out in May this year. The backbone, they use RasNet 50 anyway. Also, we put the deep learning side by side. One is recognizing the global view, the other one is local view. That means we have the zoom out and the zoom in, view as well. And also, we do the transfer learning, to try to get the best possible result. So, I'm going to show the result next anyway. This is the feature importance evaluation. As what you can see here, all the features are roughly about useful. So we don't get anything which is totally not useful, or we don't get anything which is totally very useful. So in other words, we use all of them. And feature selections may be our next step.
Yeah, Dr. Simon, we cannot hear your voice now.
All right.
Okay,
you can hear me
we
now?
can hear
Okay.
you now. Okay.
Yeah, kind of on and off anyway. So important thing, I repeat twice or maybe thrice. Make sure that you can hear me. Anyway, we need some ground truth because we are not just outputting yes or no, cancer or not cancer. We want to tell we mark this the part that that has a problem, because nowadays the endoscope may come with a little pilot, a little tool that actually plug a bit of samples, from the area. So make sure that we plug at the wrong place. Okay, so we are comparing our deep 3. The top are the result from deep 3, and the bottom one is UNet. These two are the one of the most popular deep learning algorithms that we have so far. So you see that the red border is a ground truth, and for the DeepLab 3, even though it can't fully, I mean, it can give us some reasonable result, especially the one on the most right side, green and the red are matching. Compared to UNet isn't very good. Here, there are actually tiny dots scattered beyond that red circle, and in the middle pictures, it actually miss out. It can't show at all. And the last one is actually a bit messy, as you can see that a lot of blue in some weird shades. All right, so one more anyway, one more. The red and green are good, even though they are not perfect. There's still some room for improvement, like here, the one in the middle. The green circle actually is outside. It should be inside, but for some strange reason, it moved outside. And below here is worse. So the conclusion is UNet is not as good as the.
Yeah, we lost your voice again, Prof. Simon. Sorry. Yeah, we still cannot hear your voice. Yeah, I'm sorry, we cannot hear your voice now. Maybe you can turn off your camera again to help the streaming get better. Yeah, I'm sorry, we still cannot hear your voice, Prof. Simon. Yeah, we can clearly see your slide, but we cannot hear your voice. Maybe the device for the sound, you can yeah, we still cannot hear your voice. Maybe you can fix the connection of your audio device. Maybe the cable or the plug-in need to be fixed.
Okay,
maybe. Okay, we can hear your voice now.
Okay, when I stop sharing.
Yeah. Yes, you can. Yeah, we can see your slide clearly, but we still cannot hear your voice. I guess the connection of the audio device not properly plugged in, maybe.
Yes. I
Okay,
thought here is
we can hear
Wi-Fi.
you. We can hear you
Yeah,
now. Okay.
I just disconnect and connected.
Okay. Okay.
Hopefully it's
Please.
good. Yeah.
Yeah.
So
Hopefully
this
it is
is
good now.
actually telling us a message. SCDS 2022, let's please do it offline. We don't need Wi-Fi
Yeah.
anymore.
Okay. Okay. Please
I
go
want
on,
to
bro.
be face
Yeah.
with you.
Okay.
Yes, with yes. This is the end. Now questions. Okay, maybe I make a conclusion. The conclusion is we have a very challenging problem of dynamic images, and we will try everything. Textures, segmentations, and we found out actually how we cluster the spot is very important, and that's why we try different clustering algorithms. There are many factors anyway: segmentations, clustering, and also deep learning. So we try UNet, we try the Deep Lab version 3, and they give us different results. So how do we get the best possible combinations of soft computing tools, all right, in order to achieve the best result? So I think that is everybody's questions, not only me, but anyone who do this medical deep learning. We want to achieve the highest accuracy. So this is the end of my presentation. I hope that it doesn't, we don't stop here. We would continue the collaborations. We could work together and we can come back again as CDS 2022 in Malaysia, offline, and no more virus. Thank you.
Okay. Thank you very much, Associate Professor Dr. Simon Fong, for giving us such informative and interesting presentation. Even there is some small technical problem, but we can see clearly from the slide you saw that, yeah, it is quite interesting because you demonstrate some application, and actually, I think we can follow your presentation, even there is some noise problem. Okay. Now, ladies and gentlemen, now come to the question and answer session. I invite anybody to give question or giving some remarks. Okay, please welcome. You can raise your hand, please. Okay, there is a presenter.
Yes.
Zuraini Othman. Okay, please.
Okay, can you hear me?
Yes, I can hear you perfectly.
Okay, my name is Zuraini Osman. I'm from UC Technical Malaysia, Melaka. Just want to hear your opinion about the first phase or just now your research just now, which is the first phase. We usually mention about pre-processing phase, right? Okay, in your opinion, how important pre-processing phase to help your proposed work just now, which is involving in the deep learning? Okay, how important the pre-processing to help your system to be more accurate? Okay, just want to hear your opinion. Okay, thank you.
Right. Of course, it's absolutely important in every application. Pre-processing, even the images, the quality of images, what you train, just like how you teach a child. If you teach them the right material, the child will be good, and vice versa. So in my opinion, there's a few things we can do. One is we would, first of all, somehow use our judgment to make sure that the quality of the data, the images are good. No irrelevant data, no redundant data. And the second thing is, of course, if the case that I had before, which is not just about cat and dog or apple and orange, we are trying to characterize a very specific small area that may be a lump or maybe a small tissues or maybe ours. So in that case, in my experience, the clustering algorithms is absolutely important. And unfortunately, there is no perfect. I mean, if you play with k-means, you know that it will never be perfect. But we try to get an algorithm that is perfect because the shape is very irregular and it's fuzzy. The difference between normal and not normal. You know, maybe the center part is very abnormal, but the surrounding part look very normal. You know, so the clustering algorithm, we try to use something fuzzy. I think fuzzy C-mean was a good choice, common sense, and with all these possible algorithms that you have used, I suggest you can try also the data augmentation. That means the same image that you think is already very good, but you would simulate more of it, like you're turning the angle, you rotate it, or you adjust it with different brightness. You know, that will help the deep learning. Yeah, this is my opinion. Yes.
Okay, thank you so much.
Thank you.
Okay. Yeah, I think quite interesting question and very good answer from Professor Fong. I invite another participant or even another speaker or committee to, okay, this Prof. Jasni Muhammad Zain, please give your question or some remark.
Hi, Dr. Fong. Can you hear me?
Hi, Jasny. I can hear you perfectly.
Oh,
Now
okay.
it's good.
Thank
The
thank
PowerPoint
you
is
very
causing the
much.
problem, the traffic jam.
Thank you very much for your very
Next
nice
time I talk without
presentation.
PowerPoint. Yeah.
I just want to know about I mean the process. You must be working closely with the surgeon or medical doctor to be able to interpret the dynamic image like endoscopic images, right? I mean, it's not an image; it's a video. So, yeah. I mean, how does the surgeon help you in the process of getting the information that we need? Thank you.
Thank you for the questions, Janice. Well, you are very right. I am a PhD. I'm not MD. I know very little about disease, actually. How I do this, we pay them. They won't work for free. So we start off with a project with the budget anyway. Some part of the budget is for annotation. Annotation means we will require some expert to give us some ground truth. And this kind of ground truth, unfortunately, is still manual. That is, we need somebody who's qualified not really side by side.
So you off your video. Oh.
You can turn off your video, Prof. Dr. Simon Fong.
Too much video dia akan.
Okay. We cannot hear your voice, Dr. Simon Fong.
My video is okay because dia.
Yeah, it is in the Prof. Simon Fong side. I think the bandwidth is low at the moment there. We still cannot hear your voice, Prof. Simon Fong. Yeah, as mentioned by Dr. Simon Fong, that the medical doctor or surgery is, yeah, they paid for that because there is no work for free. Of course, if we have grant, of course we can pay them using our grant, right? Yeah, Prof. Jasni. Okay, we are waiting for Dr. Simon Fong. I think he is trying to make the network work. We still cannot hear your voice, Dr. Simon Fong.
All right, coming soon. Now,
Okay.
good.
Okay.
Now, good.
Good. Now good. Okay.
It was a demonstration. We need to
Okay.
pay for the mobile data, then have good connection. Free Wi-Fi doesn't work. We pay peanut, we get peanut. Anyway, back to the question just now. Yes, annotation. So it could be systematic because we plan a schedule about by when we need the samples to be annotated, and therefore we could move ahead. Without the annotation, we can't move ahead. Yeah, so the doctor are also usually are very busy. They need to be told in advance. You got to book them like 2 weeks in advance, maybe 1 day, 8 hours. They need to do all these, and they will tell you how many images they can annotate. So yeah, I think it's an important part. Unless some doctors will do it for free, but at the end they may, I don't know, they may want to be the corresponding author. Yeah, it's a collaboration anyway. I don't mind. I don't take people for granted, all right? So it's a collaborations, and it's win-win anyway. We need to answer your question, yes, I can't do it myself. My students also can't do. We are from computer science, so we need their medical knowledge to help.
Thank you. Thank you so much.
Okay.
I should use mobile data. Yes.
Yeah. Okay. Thank you, Prof. Desni. Okay. We are finally come to the end of this session. We say many thanks. We appreciate for Associate Professor Dr. Simon Fong to give such a great presentation and also very fruitful discussion. And then, ladies and gentlemen, we give applause for the speaker and for you all. Okay, we can give applause, and also there is some reaction in the Zoom that we can give to Dr. Simon Fong. Okay, thank you very much. I give the time back to the MC. Please, Maura and Sekar.
Thank you. Take care. Thank you.
Okay,
see you again hopefully next year.
See you.
Yeah.
See you.
Yes. No virus, no mask. Okay. Thank you.
See you. Thank you so much.
Thank you. Thank you.
Thank you.
Thank you to Associate Professor Dr. Simon Fong and Dr.rer.pol Dedy Prastyo. And we would like to give Associate Professor Dr. Simon Fong a certificate of appreciation as our gratitude for your time. We sincerely enjoy your speech, and everyone, please give us your thumbs up and applause reaction. We will take a photo with Associate Professor Dr. Simon Fong, and I will count to 3, and please to the committee to take the photo. 1, 2, 3. Okay, thank you so much, Professor Dr. Simon Fong. And now we're going to have a photo session with all of the participants. To all of the participants, please turn on your camera and give us your best smile. And to the committee, please take over the time to take the photo.
Okay, for 1, 2, 3. Okay, next slide. 1, 2, 3. Okay, for the third slide. 1, 2, 3. For the last slide. 1, 2, 3. Okay, thank you.
Thank you so much to Professor Dr. Simon Fong and all of the participants. And ladies and gentlemen, after this, we will have a 15 minutes break. Please come back at 10:55 Indonesian time and 11:45 Malaysian time. Thank you.
Thank you, Prof. Simon.
Thank you,
Thank you.
Prof.